Yeah, we're right up at the 4 o'clock hour. We can give another maybe minute or so.
Yeah, I think we can get
Yeah.
started with that.
Yeah, why don't we get started? It's 4 o'clock. We like to be punctual, and I'll promote them once I see them. So welcome everyone to another edition of Tumor Talk, the collaborative effort between Lenox Hill Neurosurgery, the Journal of Neuro-Oncology, the tumor section, where we talk about recent publications in the Journal of Neuro-Oncology. We highlight with the authors their rationale for the study, their results, and probably most importantly, the relevance of their work. Today is another fantastic program. We've got an educational grant by the company Omniscient, which is a company that focuses on connectomics, which has been nice enough to sponsor a few of these presentations now. And we're joined today by Dr. Sugrue, who's going to talk to us about a recent publication in the JNO. Before we start, I do want to take a couple minutes and just introduce Ben Cobb with us. Ben is, he'll introduce himself, but he's functioning as kind of the lead on a program called Cassini, which is we're going to migrate the Tumor Talk webinar series over. It's brought to us by Springer, and Ben will tell us a little bit more about this. We think it's going to be a really valuable collaboration, allowing people to better subscribe to what we're doing, get better updates. It's going to make the process more efficient. And Ben, why don't you, go ahead and introduce the kind of what we're doing and why we're doing it.
Great, thanks so much, Randy. So hi, I'm Ben. I'm one of the co-founders of Cassini, a platform that makes it easier for researchers and journals to run impactful seminars. And we're working with Springer Nature Oncology, and particularly with the tumor talks, to make it easier to find the talks after they've been given, and to make it easier to subscribe to updates. So what I'll really quickly do is I'll show you what it's going to look like and where you can find the tumor talks going forward. So going forward, you can actually just scan this QR code. I'll come back to it in a second. So you can scan the QR code, but here's the URL at cassini.com/s/tumor talks. And when you go to that page, what you'll find is something that looks a bit like this. And you can just click on that blue button, and you'll automatically get notified of your upcoming webinars as they're coming up. And you'll also be able to get notified of recordings for webinars to make sure that you don't miss any. And here, if you scroll down the page, you'll see an archive of all the past webinars. And you'll see that each one of them has also been assigned a DOI. So if I just click on one of those and give an example, you'll see that these are citable talks. They're connected to the scholarly literature through this DOI. So they're going to be there. You can come back to them anytime. And we've also tried to make them as convenient for you to come back and watch as possible. So if you don't have time to watch the whole thing, you can actually jump right into the slides that you find relevant. You can skip through the slides, find a bit that's interesting. And we've also done is we've produced an automated transcript for each of the talks. And we've also indexed the content of the slides. So you can go ahead and you can search for terms inside this webinar. So for example, if I search for the word eloquence here, you'll find all the times that either the speaker has mentioned the term eloquence, or times that this word appears on the slide. So if I go here and click on this, you can see that here on the third slide, this is all about eloquence. And you'll see Cassini is even picking up on references contained in the slides. And you can go ahead and you can click on the link and be taken straight out to the journal article. You can see in this case, it's an article right from the Journal of Neuro-Oncology. So just in case anyone missed it, I'm just going to go back to the first slide I was showing you. You just make sure you capture that URL, cassini.com/s/tumor talks, and you can subscribe there for updates of future events. So
great. Thanks so much, Ben. And there's going to be a lot more information about this coming forward as we transition the platform over there. Jason, do you want to comment on the role with the journal and the support from Springer for moving forward with this?
Yeah, I'll just say one, thanks for joining, Ben, and I want to thank Springer and Cassini for allowing the Journal of Neuro-Oncology to be one of the early embracers of this platform. I think it gives us a lot more visibility, a lot more searchability and use of that content that we're generating. And I'm particularly excited about the DOI that's assigned to all the talks, so that this is something that can be used for easy access, for placement on your CVs, even potentially arguments for promotion tenure as you move forward in the academic world. That's an important part. These talks are now going to have that visibility, much like a publication would. So Thanks again, and excited about this as we move forward.
Yeah, thanks so much, Ben. We know it's late there in London. So really
Thanks, Ben.
appreciate it. And now we can get back to our regularly scheduled programming. Mike, do you want to go ahead and reintroduce yourself and we can dive right into the talk? I'm excited to hear what we're talking about today.
So I'm Mike Shagru. Probably some people know me. And I'm a neurosurgeon, founded Omniscient, co-founded with Stefan. And I've been spending many years working in the area of connectomics, and particularly how do we utilize this in clinical practice. And this is a topic of in connectomics and in the running series of what's called graph theory. We'll talk a little bit about it. It has a very complicated title, and I'm going to try to make this approachable because, you know, if you get past the title, it's actually fairly straightforward what we're discussing. And ultimately, what we're trying to get at is, and it's the same thing we've always tried to get at when we talk about neurosurgical topics, which is how do we deal with the fact that every part of the brain is doing something, but still make intelligent decisions. We know we can cut parts of the brain and people go on and live happy lives after that. But that doesn't always happen, and there are consequences. And particularly as we begin to raise our standards and shoot for a higher bar, which is essentially that it's not just a minimal, viable brain that we need to save. It isn't, speech and motor, and then everything else is kind of irrelevant. We know that we cause cognitive problems, but we don't know how to avoid it. And what we're really trying to do is figure out how do we get at this question of figuring out what is essential before we cut. So we've discussed a concept called graph theory. Graph theory is essentially how do we take a very complex network, reduce it to a gigantic math equation called a graph, which shows all the relationships of all the parts and how they interact, and then begin to run experiments to learn how the graph works. That's basically graph theory in a nutshell. It was really not something people cared much about mathematically. There was a few people working on it in the 70s and 60s and things. But what really made it take off were computers, and more importantly, the recognition in 1998 that the graphs of multiple types of networks that had nothing to do with each other had similar mathematical properties. So, for example, the internet and the brain have similar math. One of the things we discussed in previous ones is the concept of hubness. Okay, so hubness is the idea of how information flows in a graph, with the idea that certain areas are really connected in this, they must be important. But how do we actually figure that out? And so we've discussed in the past that hubs tend to be eloquent areas. But how do we make judgment calls? Because it's a good thing to know that the motor cortex is a hub, and this is a useful tool. But how do we know what the relative consequences are? And how do we know that if there are some areas that may not be hubs, nevertheless, they may have more indirect or subtle ways because we're often removing multiple parts of the graph in one operation. How do we dive into this? Again, we know that this is a problem. And what we're really trying to look at is the idea of this. If I remove a node, can the signal get from A to B? Now, for a long time, we've looked at temporary deficits and said that this is swelling. And, to some extent it is, but we know that swelling goes away in a few days, not 6 months. And there are people who get benefit and recovery up to, even past a year. So we know that that is not swelling hanging around for a year. That's just unrealistic. What we know is that that in many cases, even if we remove a node, the other areas can upregulate through long-term potentiation, change their synaptic weights, and the signal can go around it. So, but how do we figure that out? Because we know that that isn't infinite. We know that we can't cut we can't cut out certain tracks and function and guarantee that that's going to be true. So if we remove that signal, how do we know that the signal can get around? And that's a really complicated question. And this is something we're still learning at. What we were exploring in this paper was looking at different types of centrality. So in the past, in the previous work, what we were looking at was variations on what's called degree centrality. Now, we use one called PageRank, but essentially what degree centrality starts off with saying that if 3 things connect to you, you're more important than if 2 things. And if three things connect with more fibers, are more important than three things with less fibers. And again, there are different ways to look at it, right? So if you look at eigenvector centrality, well, it turns out that that even though both of these guys have a degree of 1 on that, if you look in that cartoon, there's one guy who basically is connected to a more important node than the other guy. Okay, so the red one's more important. And PageRank goes further, saying that, well, just because you're connected to one important person, you're probably less important than if you're connected to two important people. Okay, and so ultimately, again, these have different properties. And what we were interested, first, we were just trying to just do what we do all the time, which is just say, well, people have studied these a lot, and in the literature, there's different ways people do it. There are actually even other types of centrality, one called betweenness, where you look at every single short path in the brain, and you figure out how many go through that particular area. And what we found when we were studying all these is that most of them were either variations on eigenvector or PageRank. So even though they might be calculated differently, they strongly correlated with one or the other, but these things weren't the same. So ultimately, we make graphs from DTI in these studies. And what you do is you take a diffusion image, you put a parcellation scheme onto it, and you calculate how many fibers go between everything and everything else, and you get a giant matrix. Now that matrix is equivalent to a graph. So you can see that little diagram on the brain there. It looks beautiful, but it's impossible to study this with the naked eye. And so you really have to use mathematics to calculate what goes there. And ultimately, if you look at the group level, okay, these things roughly correlate. Okay, but you notice that they're not all along a straight line. There's some variation into it. And ultimately, what we were interested in is not the fact that these were kind of the same. We know that centrality measures should all somewhat correlate. We want to know where they were actually different. So we didn't care about that zone. We cared about the edges, particularly where they mismatched. And it turns out there's a fair number of areas that mismatch at the top and bottom. Okay, so if you look at this this line, ultimately, if you say who are the ones that mismatch, in the middle ranked mismatches, meaning high PageRank and low eigenvector or vice versa, the ones in the middle are just completely a random scatter plot. They're not consistent. People have different numbers, etc. But one of the things we found was that the edges where the strongest mismatch were very, very consistent. And that was actually pretty encouraging. And so ultimately, weren't trying to answer these guys. We're trying to look at those guys, because those guys provide some insight on how these numbers are different. So if you look at the areas that have high eigenvector and low PageRank, they're mostly in the medial temporal lobe. There's a few other exceptions, the globus pallidus and a few things, but for the most part, this is the medial temporal lobe. So that's actually really interesting when there's an anatomy behind it. And if you look at the high eigenvector and low high PageRank, low eigenvector, they're not the eloquent areas, right? Because high PageRank in general tends to be really strongly eloquent. They also have high eigenvector. The mismatches tend to be the CN and DMN, the lateral part of the hemisphere, and also some medial portions as well. And so what we try to get at is, why is the medial temporal lobe this mismatch? So we looked at another measure called clustering coefficient. This is pretty interesting because it's measuring bypassability. So if I'm that blue node and I want to look at the clustering coefficient, I want to know, if I cut that out, how easy can you go around it? Well, this one on the left has a low clustering coefficient, because if you take it out, the whole graph falls apart. Whereas this one here, there are triangles that bypass it. And so you can calculate how, if you take a set of nodes around that area, how clustered are they? How much are they interconnected where you can just ignore it? And when we measured this out, it turns out that people with high EP difference, the regions that have high EP difference, are also the ones that cluster. And we can get into the basic rank of this, but in short, the bottom line is, if we have a low EP difference, it means that the PageRank is important, and the PageRank defines everything. And this is a low bypass. It's not a bypassable area. So if your PageRank is generally high and your EP is high, or just if your PageRank is high in general, then that's bad because you take apart the graph. But if you happen to have an area that seems a high eigenvector, but a low page rank, it's actually telling you that area is bypassable and you can go around it. And again, what we need to eventually do is start to test this clinically. But we also know that the amygdala and hippocampus tend to be areas of the brain that you can remove, at least on one side, and often they're tolerated. Now, we know that that isn't always true. And this, of course, raises some questions of the age-old question of, well, can hippocampal load handling and can the patient cognitively handle a hippocampectomy? Well, this is one way to answer that, I think, and it's something worth investigating. But what we really have learned from this is the nature and property of these features. How do they behave? Where do they behave this? And we found some consistent patterns that show something that might be interesting clinically. Thanks.
So super straightforward, very easy to understand. Appreciate it. No, listen, I think fantastic work as always. It's really amazing how the software and really the incorporation of connectomics into the clinical practice can really change how we look at stuff we've been doing for 100 years. Can you go back a little bit? I want to be clear because not all of our students are mathematicians. Not everyone's a student, but not all of our viewers are mathematicians. Clinical correlates of this, right? So you talked about the medial temporal lobe. You said that this is in general a resectable area in some. What do you think makes the difference here? What is it that changes the page rank in a specific person or the eigenvector rank in a specific person?
Well, what you have to remember about getting into network theory, right? So let's go back to this graph, right? So this is conceptual. Now, what happens is that in general, the lines between areas are reasonably consistent. There's some exceptions like frontal lobe, temporal pole, that tend to be a little more variable. But for the most part, we're not all special snowflakes. Most things, whether or not there is a connection between area A and area B, most parts of the brain have at least one connection, at least in the ipsilateral hemisphere. Having said that, there are variations in that, and more importantly, there's variations on how strongly things are connected. And the reason is because the brain is a self-organized system. So there's a lot of rules that are set by genes and also by energy handling and other things, just the physical constraints of the head. And so what happens is the brain organizes it wires itself in certain ways. Now, the other thing about it is because of where the visual and sensory and all that inputs are, there's only certain parts of the brain that get connected to the outside. This sort of drives the organization of other areas. So it's not an accident that the motor planning areas started very close to the motor system, because that's who talks to the outside. But so what we know though is it's striking, given that there's a lot of just, well, the rules are set and then the playing field happens however it happens, how consistent human beings are. But we also know that wiring is not 100% consistent. People have different wiring. And there's a lot of early emerging evidence that's starting to point to the fact that in relatively young children, you can begin to see changes that happen over time that predict the future incidence of mental illness. So we can start to see patterns that happen in kids who are going to be mentally ill teenagers that don't happen in other kids. And of course, that's obviously really exciting. But what we're really looking at is that variability is very difficult to see with the naked eye. So we're not going to pick this up on our own. We're not going to pick this up by having computer programs that make pretty pictures. We need to have numbers, and we need to be able to run experiments. And so this is one effort to try to get at saying, well, okay, we know that this is in generally true in cohorts of individuals. What we're really looking at is, okay, and then when we get to the individual level, does it have predictive value? Does this tell us differences that we care about clinically? And because ultimately, if this number between these, the purple node and the node below it is 5 or 10, does that matter? And the answer is these are extremely complex relationships. And the nice thing about it is computers can do these calculations in milliseconds, right? Or depends on how big the graph is, but it doesn't take very long. And as we begin to figure out what these things telling us, they start to align with properties that we care about. Like we know that the medial temporal lobe is often resectable, right? And people, if you do it on one side, people are okay, right? It isn't the same as the motor cortex, but there can be consequences. So how do we figure out that subtle thing ahead of time by saying, well, turns out this person's hippocampus isn't as good. So you have to weigh whether that's a good idea. And I think that the only way we're going to do that past the understanding of the basic properties of the measure is empirically. Like we had to figure out what the normal range of potassium was, but before we figured out what the causes of abnormal potassium and the consequences were.
Sure. Well, that actually brings me to kind of the next, I think, logical question, which is, we always worry about the clinical correlates of these things or the clinical relevance, right? So how does your guys' work, I guess, or how does the software see incorporating this into how it informs a surgeon? Is it that, these are, necessary studies on everyone? Does the software in its current form actually evaluate this for you? Or is this, an interpretation?
At some point it's going, well, yeah, eventually what you can, it's actually fairly straightforward to calculate this, right? And it's calculated in the back end, and we use it for other reasons all the time. It's actually how QuickTone makes its atlases or graphs at the end of the day. What we get to is that it all comes down to, number one, reproducibility. How do we get tracks that the numbers are reliable regardless of what scanner it's on? We think we've mostly solved that problem. And then the second, of course, is how do you display a graph with 360 nodes that everyone can figure out what it is? So we've thought about a lot of ways to do that and incorporate it. I think one of the simplest, applications of graph theory is what Jackie showed last time, which is unexpected hubness, which is that you don't need a software to tell you that there's the motor cortex is probably a hub. It almost always is, right? But it's knowing that there's a hub where you don't expect it. And that's useful information because you might think about taking a different angle if you knew that this isn't a one-size-fits-all approach. This is kind of the next level of that, which you would think about it in the terms of bypassability, or you could calculate clustering coefficient, and all these things are kind of telling you different views of the same thing. But essentially what we're looking at is that if I knew that, yes, this is kind of a hub, but actually you can go around it, then that's useful information, because that means that you can cut it out or not cut it out. Remember, you might find out that the temporal lobe, this is not a normal hippocampus, and this is going to have consequences. Ultimately, we need clinical data behind these. What we were trying to do with this is to ultimately, people have done a lot of really interesting science with graph theory, but no one's really spent a lot of time figuring out what parts of the brain are and what does this potentially mean. And that's what we're doing. But the next step, of course, is, let's collect neuropsychological data with people and figure out what does it matter,
Yeah. I think that's a quite an important observation. So I said more on the technical side of the software here. But PageRank is something that has been around for decades. One context you might have heard about it is how it organizes the information on the internet. This is the algorithm that made the success of Google initially. And so aside from the findings that we're discussing here, what this illustrates is that now from a brain scan, we're able in a very rapid amount of time to organize the brain in a graph. And not only are we able to do that, but we're able now to manipulate this graph in a very abstract manner, so we can extract those findings pretty easily. And those findings have a potential impact on understanding better patient outcome. I think that's a true revolution, being able to do that in such a consistent and quick manner, which will ultimately push the field beyond what it is already.
Yeah, sorry, go ahead.
No, I was intrigued by this. Obviously the talk is phenomenal, and the approach is potentially revolutionary. You would, I think, talked a little bit about the clinical validations. And so some of these things are going to require some very detailed neuropsychological and cognitive tests. So can you just share a little bit about what that looks like in terms of clinical validation, what things are already underway, where you're planning on taking this next? It's an incredible tool.
Yeah, so there's a project right now that we're helping to get off the ground. It's not directly sponsored by us because obviously answering the human brain is not an industry question, it's an academic question, but obviously our tools can help make what would be very difficult to do fairly straightforward. So it's called the Glioma Connectome Project. There's a couple sites, Miami's and Penn are kind of running it, but there's many other groups that are involved in it. And ultimately, that's one approach. And that's really what you're getting at. It's large scale, detailed neuropsych, longitudinal follow-up, et cetera. And that's going to be the first starting point into this, right? But that may take a while. And also, one of the challenges we always know is we don't know what the statistical power necessary it is to find things. It may be really obvious, or it may be that there's very subtle relationships, or it may be that there's only a small number of people who have this variation. So what we, ultimately the longer term is, and this is kind of our view, and when it really dawned on me what it would take, I realized this isn't going to ever happen at a single center, right? We all kind of need to be pulling on this in different ways, because over time, there's relationships in the brain that are probably really important that we don't know and wouldn't be able to solve by just collecting our data over 10 years and then writing up a report of 75 patients. So I think what it gets down to is we view it as one of the mandates as we go forward is we need groups and registries looking at different topics, because the era of this is how I do it, and here's my, we need to have consortiums working on problems, because the problems left are not going to be solved by here's my master series of 25 years experience. It just, we've done everything we can with that, I think.
I think your strategy sounds perfect, and obviously, I've had some interest in registries and efforts, and I agree with you wholeheartedly that any single center, even a busy center, as comprehensive as they may choose to be, is probably not going to be sufficient. So it's marvelous. I was aware a little bit of that that group that is being organized. So that's a great step, and I think our audience should could find a little bit more about that, hopefully by searching about it. And I know a number of those leading centers are already engaged in it. So fabulous.
Yeah, and to answer the question in the Q&A, how would you ethically get solid clinical data? I think obviously you're not going to run this as a randomized controlled trial, right? And I think we have to get to the idea that a lot of questions we face are not really amenable to good RCTs. What we've approached it, we say, everyone just do the surgery the best way you know how. We don't know whether graph theory should or should not be used to guide a surgery. It seems like probably yes, but we don't know exactly what to do. So what we do is we say, let's have people do surgery like they've been doing, and then we learn from our own patients, right? I mean, the best neuroscience research labs in the world are our hospitals, and we don't really go to that lab. We try to do it in some awkward way where it goes off to some research group. And we really look at a very small percentage of our patients. So what we looked at with the GCP, and what I think we really need to start going back to was some of the greatest discoveries in neuroscience that have guided thousands of R01s at this point are like someone studying their patient after a temporal lobectomy and realizing, hey, bilateral temporal lobectomies is bad. And so that's how, ultimately, that's a really valuable tool, and we've kind of got away from it. Or to the extent that we do it, we mine it out to some other group, as opposed to really integrating it into the hospital. And so a lot of the most interesting patients don't get studied, right?
Yeah, absolutely. Guys, I want to thank you again. We're on the 30-minute mark. I do appreciate your guys' time. Fantastic study. I think you guys have laid the foundation for, like Jason alluded to, something potentially very revolutionary in terms of what we do. We're happy to have you guys on board, and we want to thank the Omniscient Company for the educational grant to provide these 3 webinars. They will all be uploaded to the Cassini platform as we move over to there. And we look forward to having you guys contribute more and coming back, as the Glioma Connectome project unfolds and we get some answers back. So.
All right, sounds good. Thanks, guys.
Thanks.
Great,
Thank you
great
much.
presentation. Thanks for joining.
Thank you.
Have a great
Bye-bye.
afternoon, everyone. Take care.